So now I'm going to try to give a short 20 to 30 minute talk. Maybe it'll go a little longer. We can have lots of Q&A, but I'm trying to, oh, okay. Susan Dettinger just sent to me, Q&A is only available in the quote webinar in Zoom. So we're going to just use the chat. Thank you, Sue, I appreciate it. Now I'm going to see if I can fire up my PowerPoint. While I'm doing that, are there any other questions? Because what I'm going to do is put us all on, oh, looks like everybody went to, pretty close to everybody went to muted. If you could mute yourself, that would be great. Paul, if you want to be an exception, that'd be good, because then you can talk to me, and same thing with Kendall, and that you can coach me. And remember, when we do this the other times, we're going to be having a speaker who's giving a talk from their house and may not have done this before. So this is going to be a little clunky until we all get really good at this. Oh, you want to do that? So now I'm going to hit share screen or not. What are you folks seeing now? Paul and Kendall, are you seeing my?
I have all the pictures. I have all the individual pictures yet.
Okay.
Yeah, we don't see your screen.
All right, so I got to end show. End show. Now I have to hit share screen. This one. All right, so now what are you seeing?
Now we see your screen.
Yay. And now I'm going to hit this. Okay. And now what do you see?
PowerPoint.
Science Savvy with Food Experiments, Tom Zinnen.
That's the wrong one. Happy, happy, happy day. I'm trying to figure out how to get out of this. Show. All right, how's that look?
Biotechnology and food updates.
Okay, sounds good.
That sounds like your title.
Okay, so many of you have heard me give a talk from time to time. I'm going to emphasize biotech and food things that have happened in the last 5 years, and a couple of stories that are just happened this week so far. One is on BT corn registrations, and the other is on the virus of white mold, the fungus of white mold that seems to knock down the white mold itself when it's infecting other plants. So let's see how this is going. One of the things I love about my job is that biotech and food is a profound issue, and that's because it affects so many things about our lives, our families, our bodies, our environment, our view of what's right. And I've been doing this job, I'll start my 30th year on the 3rd of June of 2021, and this is what makes it interesting. These questions I ask all the time, but they're really pretty germane for the last 6 months. Especially COVID, but also what's been going on with other social and public issues. So what is science, and how is science different from other ways of knowing? It's a pretty interesting thing to get people to think about. How many different ways do we think we know stuff? And when you rank those different ways of how we think we know stuff, where does science come out? For some of us, science is at the top. For some of us, science is down in the middle. For many of our fellow compatriots, down pretty low. And I have to be conscious of that whenever I'm talking with people about science, because I make a scientific argument. And if they don't think that's important, I brought the wrong kind of food to the buffet. So this branch of philosophy that asks what is knowledge, what is proof, is called epistemology. And I make a little wordplay out of that by capitalizing the STEM. For the last 20, 25 years, STEM has been the acronym for science, technology, engineering, and math. I like to point out that it started off as SMET, and some genius decided that STEM sounded better than SMET. So, as a puzzle question, what is food? Can you name 3 foods comes from stuff that's not alive? And usually I do this as a build and the Twinkie comes up later, so there's a little visual joke there. This is news to some people, not to tons of people, but to some people, they think there are things that humans eat that have not been alive that are foods. And it's a pretty tough thing to come up with. Sometimes people will say things like salt, and I'll say that's true, but if you run outside in July and work up a sweat and then lick your sweat, you'll find that it's salty. So even salt comes from things that have been alive. If anybody can come up with an idea of a food that comes from stuff that's never been alive, let me know. So nearly all our food comes from stuff that's been alive. Actually, I think all our food does, but I'll put a little wiggle room in there. And those things include plants, animals, and microbes. And in the last 10,000 years or so, humans have developed a thing called agriculture. Before that, we had hunting and fishing and gathering. So in the last 10,000 years or so, humans have selected and developed crops, livestock, and cultures. So plants, animals, and microbes, stock, livestock, and cultures are the domesticated versions of those. And we select those crops, livestock, and cultures on the basis of a lot of different traits. And among those are taste, and color, and ease of preparing, the yield, their vigor, their nutrition, and of course the big one is taste. Where do traits arise? Well, traits from a biology point of view are a function of their genes and their environment. That means if you want to change the traits, you can manipulate the genes, you can manipulate the environment, or you can manipulate both the genes and the environment. And manipulate comes from the Latin word manus, which means hand. Same word as manager and manifold and lots of other things. So now we're going to focus primarily on plants. Much of this would also be true for livestock and for microbial cultures. But talking about plants, if you're a plant breeder, you need to have a source of genetic variation. And that big collective word for all the genes you have available is called the gene pool. And the methods that you use are to find and move and select the desirable traits. You can do plant breeding without knowing much about genetics. Plant breeders know tons about genetics, but if you've ever been to Santa Rosa, California, and gone to the Burbank house, Burbank was a fantastic plant breeder 120 years ago, but he wasn't a plant geneticist and didn't know much about plant genetics. When we talk about this, it's interesting to think about gene flow, because genes move, and that's a big deal. And that's been a pretty big deal in my lifetime. When I took freshman biology in 1971 in Dixon High School, we didn't talk a whole lot about how genes flowed. Especially now we realize genes flow within species, but also between species, and that's a very big deal in the last 50 or 60 years in biology. How do they move? Well, some of the names that go for these processes include transformation, which is when DNA just moves from an environment into a cell and the cell takes it up. Another is transduction. That's being moved, or genes get moved around by a virus particle. Conjugation is when two cells get together and move DNA back and forth between them. Cell fusion is when two cells get together and they actually fuse into one cell. And then there's plain old viral infection, which is a little bit different from transduction in the sense that it's a cell gets infected, but the viral nucleic acid doesn't necessarily get connected with the chromosomes of the host cell. So I mentioned DNA several times. It's the carrier of the genes. In the last 6 months, people have also realized, wow, RNA does a lot of stuff. I was an RNA virologist. So you got to keep in mind, because of things like COVID and flu, RNA and HIV, RNA does a lot of the heavy lifting when it comes to moving genes around in nature. For plant people, there's a bacterium. It's called Agrobacterium tuberculans, or at least that's its old name. That's what I learned it as. It's got another name, but most people still call it agro. This bacterium is pretty cool because it causes gall at the crown of the plant, and the crown is where the stem meets the soil. And one of the reasons it's at the crown is because people hoe woody plants, and if they whack the hoe onto the plant, you make a wound, and it's that wound that the agrobacterium from the soil can get into. And the amazing thing about agrobacterium is it's got a plasmid, a little circle, extra circle of DNA, and it can actually shoot that circle of DNA so it'll go into the cell of a plant, and that DNA from the bacterium will get incorporated into the chromosomes of the plant. And that means it's a vector. That means it can move, agrobacterium is a good vector for moving DNA into plants. So that's a big deal because that's what people were using starting in the 70s. Some of the first milestones in traditional biotech and food from the moving genes around point of view are these. In 1983, we got the first transgenic plants using crown gall bacterium. That means you could take a gene from one organism and move it into another. And think what that means for the gene pool of the plant breeder. In 1970, if you're a plant breeder, your gene pool for corn is basically corn and its close relatives. In 1983, when this came out, it's like, whoa, this means the gene pool is potentially any gene from any organism, any place on the planet. And that's a pretty big deal. And because you can take RNA and reverse transcribe it back into DNA, which is a process first studied by Howard Temin at UW-Madison, and he and another person jointly won a Nobel Prize for discovering that. That means even RNA viruses and other RNA things can be part of your gene pool. So the 83 was the first transgenic plants. The first ones that were tested outdoors came in 1986, and in the US, the first field test of transgenic plants was in Middleton, Wisconsin. 1990 was the first food into the American food supply, and that was chymosin. It's a cheese-making enzyme. If you're going to make cheddar-type cheeses, you need the milk, you need some bacterial cultures, you need some salt, and you need an enzyme. And chymosin is an enzyme that originally comes from the stomach of baby calves. It's a protein. But the supply would go up and down. The supply of chymosin could go up and down with how many, especially what's the word? Not heifer, but bull calves there were, because you had to kill the calf before it was 10 days old. So this was pretty cool in the sense that, wow, we can find the gene, the genetic recipe, the DNA that encodes for this protein, chymosin. And what if we move it into a bacterium? That bacterium now has the genetic information it needs to make that. It's gonna make all this, it can grow up in a culture, it can make the protein, you can collect the protein. So in the refrigerator at the Biotech Center where my outreach lab is, we have a gallon jug that still has a couple inches in the bottom of it of chymosin from 1990, and it is still active, and that's pretty amazing stuff. In 1993 was bovine growth hormone. That actually was developed in the late 80s. That's why I have a job at the Biotech Center, because of the controversy around bovine growth hormone. Bovine growth hormone is a protein. There are some hormones that are not proteins, but this one is a protein. Because it's a protein, that means you can find the genetic recipe card for it, make a copy of it, give it to bacteria, grow up the bacteria. They'll make a lot of this protein. You collect that protein and purify it. You inject it in the tail head of the cow. She'll give you more milk. Very controversial, being phased out commercially now, but this was the crux of the biscuit in the late 80s. That was the most controversial thing. 1994 was the first crop in US food supply. That was flavor saver tomato. And this is interesting because it wasn't something that was added to the tomato. It was a gene that was closed down so that the tomato could be red and ripe and taste good, but not get mushy, and therefore could be shipped over greater distances. It was not a commercial success, but it paved the way for a lot of policy and regulation questions. Then in 1995, the first transgenic fuel crops were approved, grown in 96. So that's going to be corn and soybeans, primarily for those of us in the Midwest. Eventually also cotton and canola are still the big four. 1999 got transgenic papaya, which is interesting because this is the first one that wasn't developed by a large company. It was developed by a researcher from Hawaii to help the papaya resist a virus disease called papaya ring spot. And that ended up being a big, long controversy between organic growers in Hawaii and other folks. And I think it's still on the market. Lots of cool things came out of that. The next one from 99 is transgenic potatoes were boycotted. And that's a big deal because of all the diseases in Western European history, at least, none is more important than the Irish potato famine, which is caused by this fungus, this oomycete. And in the mid-1840s, it really wrecked the potato diseases. It's called late blight, and it's still a nasty disease. Wisconsin growers, to control late blight, may have to spray fungicide 5, 6, 7, 8 times on their fields. This potato, the transgenic potato, had resistance to that, and yet it still got boycotted. And the one from 99 was a BT, Bacillus thuringiensis. It's a bacterium that makes a protein that makes the plant resist insects. And then golden rice, we'll talk a little bit about golden rice because that's a vitamin A thing. So now, in the last 5 years, what's the status of the major transgenic crops worldwide? Transgenic salmon were finally approved in the US and sold in Canada and about to be sold in the United States. This is a big deal because livestock has always been more difficult to get approved and to get consumer acceptance of. The transgenic salmon was originally first developed in 1988, and it wasn't approved until 2016. And so that's a long saga. Also, the US, the first GMO labeling law happened and passed in July of 2016. It had a very long 3 or 4 year, once it was signed, it was going to take 3 or 4 years to come into effect. There was this little thing called the election in November of 2016. And when President Trump took office in January 2017, the speed with which this stuff moved along and the rigor of it was probably different than had Hillary Clinton won. The other thing that's huge since 2012 and especially since 2015 is CRISPR gene editing. So when we talked about the first phases of modern genetic engineering, biotechnology, and plants, it was all about this idea. Can you find a copy of a gene from one organism and move it into a crop that you're interested in? This phase is interesting because now it's like, can you find a gene that you're interested within the crop, know its DNA sequence, and then go in and change that sequence, one or two or a few letters of the DNA, the A's, the T's, the G's, the C's, and get something that's valuable to you? And the answer is yes. And then the question is, how do people in the public perceive this, and how do governments regulate it? Because it's clearly engineered, but it also is not transgenic in the sense that you've taken a gene from one place and moved it into another. So, close on this. How to look at this? Well, DNA is a lot like an alphabet. And instead of having like 26 letters that English has, it has only 4. We call those letters A, T, G, and C. And so just as with English, you can take and chain those letters into words, words into sentences, sentences into paragraphs, paragraphs into pages. With English, you can do that with DNA. You can chain the 4 letters into genes and into gene clusters and into genomes. And if you change letters, whether it's in English or in DNA, small changes in the letter order can make a big difference in meaning. And then DNA is also like a box of genetic recipe cards. You can add or subtract whole cards, or you can edit information on an individual card. In this analogy, if you're moving entire cards around, especially from a DNA box of one species to another, that's the 1980s type genetic engineering. If you just edit information on an individual card and keep it in its same box, that's more like splicing, excuse me, more like gene editing. So, whoa, how are we going to look at this? And in the US, the regulators came down on, their decision was distinctly different from that in the European Union. As a former speechwriter, I always liked this phrase. I've always depended on the kindness of strangers. Only to me, there should be a line through the strangers, and it should be, I've always depended on the kindness of editors. That's a line from Blanche Du Bois and Cat on a Hot Tin Roof, I think. RNA interference, here are the different types of gene editing. One of the first ones was RNA interference, and then zinc finger, and then talons, and then CRISPR. And you don't hear a whole lot about the first 3 anymore because CRISPR is so much more elegant and easier to do than the first 3. And the reason I talked about this from being 2015 on to today is because of this story that appeared on January 1st of 2015 in the New York Times. And that is the US Department of Agriculture, which regulates crops as well as EPA and the FDA. For the USDA, the regulations of biotech crops have always pivoted on the idea that you're moving a gene from one species to another. And so you call that gene foreign. And because of that, you can raise the possibility that the engineered crop might become a plant pest. That's a big deal because the authorizing statute dealt with plant pests. We do not have a specific GMO regulation statute. We depend in the United States on existing statutes. And so it's more contorted than, say, in the EU, where they have a direct statute to be dealing specifically with gene splice crops. And since there's no transfer of genes, there's no foreign genes, and they said, well, editing the crop generally doesn't trigger concern. And that's part of the phraseology, we have no concerns, is the USDA's way of saying you can go ahead if you want to. So some of the examples of CRISPR applied to food crops: slower browning apples, slower browning mushrooms, the waxy trait in corn, which is high amylopectin. You can also tailor the number and branching patterns of tomato flowers. That can be a big deal depending on the commercial production of tomato that you're looking for and how you want to be able to harvest them. And then back to the most important thing, flavor and aroma. Those are very big traits to consumers, but a lot of times those have been not highly selected for by plant breeders. And people are thinking that CRISPR will allow us to take commercially vigorous crops that have lost flavor and aroma and restore some of that flavor and aroma. So the National Academy of Science has been putting out reports on genetic engineering since the 70s and on genetic engineering in food since the late 70s and early 80s. They do this about every 3 or 4 years. And in 2016, they came out with another report that reiterated what they had been saying for 30 years, and that is regulation should focus on novel characteristics and hazards. The emphasis should be on the characteristic and not on the technique or the process and how they got there. All technologies for improving plant genetics, whether it's genetic engineering or conventional, can change foods that can raise safety issues, and yet we don't have hardly any that I know of regulatory oversight on most plant breeding from a classical plant breeding point of view. So again, they reiterated the idea that regulations should be based on not the process used, but on the product arrived at. And that's the National Academy's advice, and that's not exactly what the US does with their 3 regulatory agencies, the Department of Agriculture, the Environmental Protection Agency, and the Food and Drug Administration. But we are more closer to that than, say, the European Union is. So here's where it starts getting a little interesting, because CRISPR was on the front of the minds of the folks that are writing this report. If new plant varieties should have these safety tests if they have intended or unintended new characteristics with potential hazards. And that is not saying only those developed with genetic engineering. This could be with CRISPR, or it could be with traditional plant breeding. And then, as I mentioned, we're more product-based risk regulation than, say, the EU, but that isn't necessarily how things go, especially with the FDA. When it came to CRISPR and livestock, they just flat out said, nah, it's still going to be regulated as a genetically engineered item. And what's amazing is the CRISPR technology is really blurring this distinction between what molecular plant people do and what traditional breeding people do. Now, the salmon. Those 2 fish look like they're very different ages, but they're the same age. One just grows faster. And it looks like the one on the left is really a giant fish, and the one on the right is a normal sized fish maybe. And what's really happening there is the one on the left is at the normal full size of a salmon. It just got there faster with less feeding. So this is the Aqua Bounty salmon, first developed in 1988, only recently approved in the last few years, and now on sale in Canada, and I think soon in the US. So that picture is an interesting one, because you could get the idea that, oh wow, this is making a gigantic fish. And it isn't. It's making a fish that grows faster to its full size, but it doesn't get to be a larger size. This is a good example of why you got to be careful what kind of images that people use or abuse in trying to tell you about what's going on with food production. So I got 1989 there. That's a mistake. Should be 1988. There's no clear route to regulatory approval for fish, because unlike crops, we haven't been collecting and breeding wild salmon. And so there was no precedent based on what to do with salmon. And it took much longer to get a regulatory system that people would have confidence in and that would be able to meet the statutory expectations. So that's a pretty interesting thing. I don't know where it's going to go in the next few years. I never do. But the FDA permitted production in the US for last year. I haven't been able to find if it's on sale in the US yet. These are grown in tanks in buildings in the US, so it's not pen grown in ocean waters or sea waters. And that's another way that they try to alleviate any concern about being transgenic and escaping into nature. Now on to golden rice. These are two types of rice there. One's yellow and has a lot of provitamin A, and the other has white and hasn't been genetically engineered to be able to make vitamin A. Vitamin A deficiency is a nasty worldwide problem. Blinds 600 or 700,000 people a year. It kills even more, several million. And to be able to get more vitamin A into diets would make a big difference in many places in the world. One way you can do that is to get a wider variety of crops, especially leafy green vegetables, into your diet. Another way is to genetically engineer rice so that it has the genes that it needs to be able to make the provitamin A. And then you have to ask, is there enough provitamin A there to make a difference? And is it biologically active when you eat that rice or prepare that rice and cook that rice and then eat it? Are the people who are eating it getting enough provitamin A to make a difference? So the first version of golden rice was released 20 years ago, had a very long process of review. The second version came out 5 years later. It had higher levels of provitamin A. The field tests for these were controversial all along. There's several cases of people vandalizing the field plots. And you can see how long it took to get this up and approved. So 2 years ago in Australia, New Zealand, and the USA, we don't depend on rice as a leading grain in our diet in those three countries, but the Philippines do. And a little less than 10 months ago, the government of the Philippines authorized direct use of golden rice. And that's both in feed and in food and in processing. So I'm watching to see how widespread this will be used, how many different varieties of rice, and there are hundreds and hundreds of varieties of rice that the provitamin A traits might be moved into either by classical breeding or by genetic engineering, and whether there's any demonstrated effect on public health because of decreased vitamin A deficiency. Citrus screening is a disease that came to the fore again 5 or 6 years ago in the United States. It's been spreading around the world. It's transmitted by an insect. It's caused by a weird kind of bacterium, and it's devastating to all kinds of citrus. So oranges and grapefruit and lemons, limes, mandarins. And if oranges are to Florida what cattle are, dairy cattle are to Wisconsin, this is how the folks in Florida were looking at it. It's like, this is threatening our entire brand name, first-rate agricultural industry. And these are pictures from UC Riverside. California has a different type of citrus industry, but both states are very tuned into it, as is Texas. This is the kind of insect, a psyllid, that moves the bacterium around. So it's caused by a bacterium. It's moved around by this flying insect. It's wrecking citrus all over the world. And there was no really good source of genetic resistance known for a long time. So people were looking at genetic engineering approaches, and that would be very cool. But then the whole question is, would people drink orange juice? Would people eat oranges that were from transgenic orchards? And then just 2 months ago, the University of California Riverside researchers announced that they had found a peptide, a protein, a small protein, in what's called the Australian finger lime. And that little protein, that peptide, seems to knock down the bacteria. It even lets the plant start growing healthy looking shoots again. So the weird thing is the plants are not genetically engineered, but where do you get this peptide to spray on the plants? Well, they took a copy of the gene from the Australian finger lime plant, cloned it, moved it into a bacterium to grow it in a vat in the lab, purified that little protein, that peptide out, and then they sprayed it on the citrus. So this is a very interesting to me way of doing this because it's using classical genetic engineering, finding a copy of a gene, moving that gene to something else, but then growing that up, extracting the protein back. So it's much more like the chymosin approach than say the flavor saver or the BT corn approach. US GMO Label Law, there you have it. Those are going to be the labels that you can expect to see starting in several months. Very different than the original ones that were proposed that looked like biohazard labels. This all looks warm and fuzzy and green, yay. And so you've already seen that people are very attuned to what you call things. Golden rice is a warm fuzzy phrase. Frankenfood is a cold, sharp phrase. People argue over words and phrases, and they also battle over the symbols that are used. So in 2018, coming up on what 21 months ago, Sonny Perdue, who's and still is the Secretary of Agriculture, announced these bioengineered food disclosure standards that were part of the law that was signed into statute 2 years previously. It defines what bioengineered foods are. And the big thing is in the US is this: do the foods contain detectable genetic material? For our purposes, that's mostly DNA. And because if you don't have detectable DNA, then under the US label laws, you don't have to label it. So for example, if you have transgenic soybeans from which you extract soybean oil, in the EU, you'd have to label that as soybean oil from transgenic crops. In the United States, you won't have to. So that's a significant difference between the way the EU looks at things and labels things and the way the US is going to label things. So earlier this year, companies were able to start using the label voluntarily. In about 15 months, they'll have to have mandatory compliance with it. And there's going to be one label for bioengineered foods, and the other one is a permitted label. If you want to brag that your food is from bioengineering, you can say derives from bioengineering. So this is a label law, but where the labels are going to be, or whether it's just going to be one of those little square-like codes on the food that you shoot your iPhone or camera phone on and go to a label, that might be how things go. So about 3 weeks ago, this was the list of foods that the USDA had that were going to be falling under the GMO labeling law. So alfalfa for shoots, an apple, especially the Arctic varieties, canola for canola oil, corn, cotton, eggplant, papaya, pineapple, potato, salmon, which is that Aqua Bounty Company's Aqua Advantage fish, soybeans, squash, and sugar beet. Looks pretty good. What you don't see here is also pretty good. This is 2, 4, 6, 8, 10, 12, 14 crops. California grows 200 different crops on its own. And what you're not seeing here are big ones like wheat, oats, rye, all the small grasses. So there's still a heavy impedance of using genetic engineering on a routine basis in many, many crops worldwide and here in the United States. So there's a pretty strong founder effect of most of these things were all done 20 years ago, and there's been very few major new uses of genetic engineering on crops that didn't make it into commerce by the year 2000. So I'm particularly focused on wheat because that's a big one. Okay. So where are we looking at? We're looking at CRISPR versus DNA sequencing and due diligence. The amazing thing is DNA sequencing is what drives CRISPR. You got to know what the sequence is of your DNA if you're going to edit it. DNA sequencing has become faster and far cheaper year by year by year. That means it's going to be plausible to expect plant breeders, no matter what processes they're using to generate variation in their crops, to sequence their crops and to do their due diligence to show that there's no significant difference or no sequences of concern. It also means that with CRISPR, it's possible you can sequence all these crops, even small crops that people didn't develop gene transfer technologies for, and be able to use gene editing without having to go through, at least in the US, some of the more difficult regulatory steps for gene splicing and gene transfer. Likewise with food labels, I don't know that CRISPR is going to trigger food labels the way that it would in the European Union, because the European Union's courts have ruled that CRISPR is genetic engineering from the point of view of regulation and labeling. Time is the idea of, wow, this first came out in 1980s. Many things were commercialized by the year 2000. We're now in the year 2020. That's 2 complete human generations. And what the generations of people coming up now think about this is different than what people were thinking when I started this job in 1991. So I don't know how time will or will not change things, but it's certainly been a long time. And then brands and boycotts. I went to a talk once where a person from the food industry said, keep in mind there are 2 huge, and he called them dirty little secrets of the food industry. The first dirty little secret was taste. And that is taste drives lots and lots of things. And he's meaning taste, aroma, flavor. The other one is brands. And that is a food company will do about anything to protect its brand name. And that gives people who are opposed to technology, say it's genetic engineering, it gives them leverage over brand name food companies that want to protect their brand, because who wants to wrangle over a boycott, even if the boycott is supported only by 3 or 5 or 10% of the consumers. You do what you do to protect protect your brand. And that particularly was used very well against potatoes and transgenic potatoes, because an astonishing amount of the potatoes in the US go into branded potato chips or into branded French fries. And finally, as always, there's always the unforeseen and the unforeseeable. I don't know what's going to happen, but it's fun to hang out. And it's 10 after 2. I started at 1:30, so why don't I stop there? And let's see if I go back. Good. So if I can, I'd be happy to take any questions. And I think you can unmute yourselves, is that correct? And I have to figure out where I am on here. So Paul, if you want to encourage people to ask any questions, that would be fine by me. And then I have to figure out how to get back. Right now I have 5 people across the top, but I don't have the rest of the.
Okay, I don't have anybody showing on mine except you, Tom.
Okay. And I'm going to stop screen sharing. Stop sharing. That's probably why. There, that's what I need to do. Thank you.
Hi, I've got a question.
We're back.
If you don't mind.
Okay,
No.
Don, this is Don Reeder.
Yes. With climate change coming along, is there a effort made to improve the yields of crops with respect to different climate features? I mean, like drought, et cetera. And, you know, would that overcome the attitude toward genetic modification?
So Don's question is, with all the climate change challenges facing us, are there efforts using genetic engineering or CRISPR to improve crops, to resist drought, deal with higher temperatures? And would that imperative help address concerns that people have about genetic engineering? I think one of the things that's going on is lots of interest in using breeding of all sorts. Whatever works is how I think many, if not, perhaps not all, but many plant breeders are going to be looking at this from. I think they're also practical in the sense of saying, wow, if we can do this with CRISPR, first of all, it might be faster and easier to do. And second of all, it has lower resistance, both from a legal standpoint and from consumer or environmental point of view for some. So yeah, I think people are looking at that very carefully. This whole idea of temperature zones moving further and further north, the shifts in rainfall. So there's lots of people looking on lots of different ways on that. And I think people are going to say, people who are in favor of genetic engineering are going to say, why take this tool out of our hands? That being said, it didn't work for vitamin A easily. Ingo Petrakis wrote a very long essay in the year 2000, 20 years ago, pointing out, wow, we did everything over the last 15 years that people asked us to do. And at the end of the day, they still opposed vitamin A rice. That's on, and he very explicitly said the ethical burden is now on the people who have opposed this, because we met your concerns and yet you keep opposing this. So I think that's one, I think we'll have that possibility in 2020 and 2040 of being able to say, wow, we addressed your concerns and yet you still are not accepting these. So I don't know what'll happen and how people will move ahead, but I'm anticipating that's at least in the playbook, and whether that play will play out again or not, I don't know. Other questions? Is this a cure for insomnia? Yes. I'm seeing the chat from Kathleen Ott, who says, I miss in-person lectures. And that was at 1:21. I hadn't even started, Kathleen. Thank you for your support. Other questions? I miss them too. They're a lot easier. It's great just to be able to show up at this place at 12:45 and have K Crew will have everything ready to go for us. Next questions.
I'd like to ask another one, which is quite off subject in a sense, and that is, I understand that bananas are endangered, and I'm wondering if genetic engineering could save them.
So the question is, he understands that bananas are endangered, and that's particularly by a fungal disease. I think the name of it would be Rhizoctonia when I was a student, but all the names have been changed to protect the guilty. So what's pretty interesting is in plant pathology, the banana that we eat today is, this is a case study from plant pathology grad school. The dominant banana, if I remember right, used to be called the Gros Michel. That was the French for Big Michael. And that particular variety of banana became susceptible to a disease and was basically lost to commerce. Bananas are propagated not by seed, but by cuttings, and therefore they're clonal, and therefore they don't have much in the way of genetic variety. And so this is a big question about how do you, if you're a plant breeder, one of the things you want is plant variation and genetic variation to go with that. And so as with the citrus screening, I'm wondering if this isn't something that people will be using, either genetic engineering, gene transfer, or gene editing to be able to address. So it's good to have bananas. I hope they can deal with the disease. Other questions? All right. Paul, do you want to?
We might want to wrap it up.
That would be good. So I hope, my understanding is because you were accepted this time, we don't have to do this next week. I will be tuned to that. And if you want to start as was suggested, start coming in at about 12:45. We can chat, we can do all that good stuff. We hope to have a speaker other than myself next week. That means we'll have to have technical connections with them so that they can share their screen and do all that good stuff. Let Paul know if you have suggestions. If you want to email them to me, that's fine. If you want to email them to Paul, that'd be great. I appreciate so many people coming on and your patience as we're trying to wend our way through this technology. Paul, do you have anything else?
Well, it looks like we had about 30 people on, so that's great. If we can thank you for your presentation. And do you want to mention anything about Wednesday night at the lab this week?
Yeah, so I'll be doing a similar talk tonight as a prototype at 7 o'clock, starting at about 6:45. I'll send to the big list of, Paul, if you can send me the latest list of people who are registered for PLATO, then I'll just turn that around and send it.
Registrations in today while we were trying to get on the air.
Yeah, so if you and Char can compile those,
I'll update it.
then I'll send that out. So that'll be tonight. It's going to be the same talk. So you may, the big thing is you'd be doing me a favor to come in and listen to how the technology is working. And then if you want to peel off once I get going, that'd be great. But Wednesday night, the lab is a little different than PLATO. We don't do the round the room thing like we did today for the first time. Paul has us do that at the beginning of every semester.
Thank you for organizing this presentation, and we'll plan to see you next week.
Thank you.
Yeah, thank you.
All right. Bye.
Thanks, everyone. Bye.
And then I'm happy to chat with anybody that wants to hang on and see how it goes. Down to the last one there, Paul. All right.
All right, thank you very much.
What do you think? Have you done other Plato classes online, Paul? Oh,
Well, we got on.
yeah. That's no small.
Yes, I have had 2 other classes already, and they all had technical difficulties, but we got through them all.
Okay, good. And are they using Zoom or are they using something else?
I think they're definitely using something else.
Are they using Microsoft Teams or?
Zoom meetings was ours, and I watched Global Affairs, which was right before this.
Okay,
And we had some difficulty getting up, but again, once we got everybody on, it seemed to go well. And on Monday, I watched Richard Steves, and that pulled off pretty well too. I'm surprised we did 3 for 3 that we actually got on.
good.
All right.
So I'm hoping John Brugge will give the talk next week. If not,
Has
I
he
gotta,
responded to you? John, are you on yet?
no. It's
So has
only
he
you and me.
responded to you at all?
He said that he had, but he might have done that in the last hour and I haven't been looking at the.
Okay, well, as soon as you know that, I think we ought to send it out to everybody.
Yeah, it's been slow. You know, I got 5 speakers, but I got 4 more that I
Yeah,
got.
the early ones are not signing up.
Yeah. So we'll see what I can do.
And if we have to cancel one for lack of a speaker, it's not the end of the
Oh,
world.
no, I'll just pull back here in my quiver and pull out 3 or 4 more great talks.
Okay.
All right.
Thanks a lot, Tom.
Thank you. And how does your wife pronounce her name?
Shar.
Shar. So
Yeah.
it's not Char, it's Shar, like Charlene.
Shar, like with SH.
Charlene. Okay, great. Well, tell her thanks for
And-
all the help, and I appreciate all that, and it worked out better.
We will carry on. Very good. I'm glad we got 30 people in here.
Yep, carry on is my favorite food.
On
All
or
right.
out.
Talk to you later. Thanks. Bye.